Hey everyone, welcome back. Ready to dive deep into some more fascinating scientific waters with me?
Absolutely. Always ready to explore the intricacies of the natural world.
Well, today we're taking a journey to the heart of it all, DNA and RNA. These molecules are the foundation of life itself.
They're the blueprint, the code, the instruction manual for building and running every living thing.
Exactly. It's mind-blowing to think about it. From a tiny seed to a giant sequoia tree, it all starts with DNA.
And it gets even more amazing when you consider that this blueprint, this incredible amount of information, is packed into the nucleus of almost every single cell in our bodies.
I know. So let's break it down. First up, DNA, the superstar molecule we've all heard about. But what exactly is it?
Well, think of DNA like a really long twisted ladder, a double helix, we call it.
Okay, I'm picturing this twisted ladder. What are the rungs of the ladder made of?
Those rungs are made of pairs of chemicals called bases. There are four of them: adenine, thymine, guanine, and cytosine. We shorten them to A, T, C, and G.
ATCG.
Got it. So how do these bases fit together on the ladder?
They pair up in a very specific way. A always goes with T, and C always goes with G.
Like a perfect match. Why is that pairing so important?
That precise pairing is what makes DNA replication possible. When a cell needs to divide, the DNA molecule splits right down the middle, like unzipping a zipper.
Okay, so the ladder unzips. Then what?
Each half of the ladder then acts as a template to build a new matching half. Free-floating bases in the cell pair up with the exposed bases on each strand, following those AT and CG rules.
So it's like each half of the ladder can be used to create a perfect copy.
Precisely. And that ensures that each new cell gets a complete and accurate copy of the DNA blueprint.
Wow, that's pretty slick. But hold on, how does all this DNA, this massive ladder, fit inside the tiny nucleus of a cell? It seems impossible.
It's remarkable, isn't it? The DNA molecule is incredibly long. If you stretched out all the DNA in your body, it could reach to the sun and back hundreds of times.
Oh, way. Seriously. But it's all packed into this microscopic space. Yes. It's all thanks to some incredible packaging. Imagine taking a very long, thin thread and winding it carefully around a spool. That's essentially what's happening with DNA inside the nucleus.
So it's super coiled and condensed to fit it all in. That's an amazing feat of organization.
It is, and it highlights just how intricate and efficient these biological systems are.
All right, so we've got the basics of DNA down. But what about RNA? How does it fit into this whole picture?
Well, RNA is like DNA's close cousin, but it plays a slightly different but equally vital role.
Okay, so they're related, but how are they different?
For starters, RNA is single-stranded, not a double helix like DNA, and it uses a slightly different sugar molecule.
Subtle differences. Anything else?
Oh, and instead of thymine, RNA uses a base called uracil. So it's A, U, C, and G for RNA.
So it's like a variation on the DNA theme.
Yeah.
Well, what exactly does RNA do?
Think of RNA as the messenger. DNA holds the master blueprint in the nucleus, but it can't leave. That's where RNA steps in.
Okay, so RNA ventures out into the cell. Why can't DNA do that?
DNA is just too precious and vulnerable. It needs to stay protected inside the nucleus.
Makes sense. So RNA is like the courier delivering the DNA instructions.
You got it. RNA carries those instructions out to the ribosomes, which are like tiny protein factories in the cytoplasm.
So the DNA is the architect with the master plan, the RNA is the courier delivering the blueprints, and the ribosomes are the construction workers putting it all together. Is that right?
Perfect analogy. And just like a construction crew needs specific blueprints to build a house, each protein needs a specific set of instructions. That's what RNA provides.
This is starting to make so much sense. So how does the ribosome actually read those instructions and build the protein?
Well, it's all about the sequence of those bases, A, U, C, and G on the RNA molecule. The ribosome reads this sequence 3 bases at a time.
3 bases at a time. Is there a special name for that 3-base sequence?
We call it a codon, and each codon codes for a specific amino acid.
Hold on. Amino acids, those are the building blocks of proteins, right?
Exactly. They're like the Lego bricks that make up a protein. So the ribosome reads each codon on the RNA, grabs the corresponding amino acid, and adds it to the growing chain.
It's like a recipe where each codon tells the ribosome which ingredient to add next.
Precisely. And since we have 20 different amino acids, the possibilities for different proteins are almost endless.
That explains how we get from DNA to proteins, but let's go back to DNA replication for a sec. You mentioned that the DNA molecule unzips, but what actually does the unzipping?
Well, it's not a tiny zipper, but there is a special enzyme called helicase that does the job.
Helicase, okay. So it breaks those bonds between the base pairs, and separates the two strands.
Yep, that's right. It sets the stage for replication.
So then those free-floating bases come in, pair up with the exposed bases, but how do they get linked together to make the new complete strand of DNA, I mean?
Well, that's where another enzyme comes in DNA polymerase.
Okay, another enzyme with a very important job, I'm guessing.
Absolutely. DNA polymerase adds the new nucleotides to the growing DNA strand.
So it's like a builder putting the new bricks in place.
Exactly. But it's also a very meticulous builder. It proofreads its work, checking to make sure each base is paired correctly.
It's like a quality control manager, making sure there aren't any errors.
Exactly. That's
Yeah.
incredibly important because any errors, or mutations as we call them, can have big consequences.
Wow, so DNA polymerase really keeps things running smoothly. All right, we've been talking about genes, but what determines which ones are active in a cell?
That's a great question. That process is called gene expression, and it's pretty complex. It basically determines which genes are switched on and which are off.
So it's not like all our genes are constantly on.
Not at all. Some genes are only active at certain times in certain cells. It depends on what the cell needs to do.
That makes sense. But what controls which genes are turned on or off?
Well, a lot of factors actually, or your diet, stress levels, exposure to toxins, all these things can impact gene expression.
So our genes are constantly responding to our environment.
That's wild. Okay, one last question about genes. I've heard the term junk DNA. What is that exactly? Is it really just junk?
Junk DNA is kind of a misleading name, honestly. We used to think these stretches of DNA that didn't code for proteins were useless, but we're learning that they actually play important roles.
So it's not junk at all.
Not really. A lot of it seems to be involved in regulating gene expression.
So it's more like the control panel for the whole system.
Yeah, that's a good way to think about it. We're only just starting to understand the full complexity of these non-coding regions.
It's amazing how much we're still learning about DNA. So we've covered DNA structure, replication, genes, RNA, and protein synthesis. It's clear these processes are all intertwined. But I have to ask, what are the practical implications of all this knowledge?
The applications are really vast and constantly expanding. Take medicine, for instance. Understanding DNA has completely revolutionized how we diagnose and treat diseases. Yeah. I've heard a lot about gene editing and personalized medicine. Are those related to our understanding of DNA?
Absolutely. Gene editing techniques like CRISPR allow us to actually change DNA sequences. It's like having a molecular scalpel that can cut and paste genes.
Wow, that sounds incredible. But I imagine there are some ethical concerns with a technology like that.
Definitely. When we're talking about manipulating the building blocks of life, we need to be extremely cautious.
A powerful tool that needs to be used responsibly. This whole deep dive has been mind-blowing. I feel like I have a new appreciation for how complex our cells really are.
I know, right? It really makes you think. From practically zero knowledge about DNA a century ago to now, where we can understand it, even manipulate it, it's incredible.
It is a real testament to human curiosity and our drive to figure things out. But we're not done yet. There's still so much more to uncover about DNA and RNA. That's what makes this field so exciting. Absolutely, and it feels like we're learning new things every single day. It's not just these super advanced things like gene editing either. Understanding DNA and RNA is crucial for things like, solving crimes, DNA fingerprinting. It's pretty amazing.
Oh, for sure. The ability to identify someone from just a tiny bit of biological material, it's revolutionized criminal investigations. And it goes beyond that too. Think about agriculture. Genetically modified crops, for example, can be engineered to produce more food, resist pests, or have better nutrients. That could help us tackle some major global food challenges.
Wow, that's incredible. We've talked about how DNA replicates itself, but I've got a question that's been bugging me. How did the very first DNA molecule even come to be? I mean, where did it all begin?
That's a great question, and honestly, it's one of the biggest mysteries in science. The origin of life, it's a puzzle with a lot of missing pieces. There are some cool theories out there, like maybe the first self-replicating molecules popped up from a primordial soup of chemicals billions of years ago, but no one knows for sure.
So it's a big open question, a scientific adventure waiting to happen.
Exactly. But even though we don't know exactly how it happened, just knowing that it did happen, that these molecules somehow assembled themselves and led to all the life we see today, that's pretty awe-inspiring, wouldn't you say?
Definitely awe-inspiring. It really puts things in perspective, doesn't it? We're all connected, part of this massive web of life, sharing a common history written in our DNA. It's pretty humbling.
Well said. And knowing that should make us appreciate the interconnectedness of everything and want to protect all the amazing diversity on our planet.
I think we've covered a lot of ground in this deep dive into DNA and RNA. We've gone from their structure and role in protein synthesis to the amazing way DNA replicates itself, and even touched on the huge impact of this knowledge on medicine, technology, and our understanding of life itself.
Yeah, it's been a fantastic journey. I hope our listeners now have a deeper appreciation for just how complex and elegant life is at the molecular level.
Absolutely. And if you're feeling like you have more questions than answers after this deep dive, that's a good thing. It means we've sparked your curiosity, and that's the first step to learning more.
So keep asking those questions, keep learning, and never stop exploring the fascinating world of biology.
Thanks for joining us on this deep dive, everyone. We'll see you next time for another exciting exploration.